Good morning, everybody. Good morning, I guess, good afternoon, whatever. And I want to thank you for coming to our we have two final DARE webinars on long-term survivors and lifetime survivors. And today our call is going to be very interesting, I think. It's going to be nothing but community people from the community who have participated in cure trials are here today to share their experiences with us. And we're going to go to 1:30 if we need to. So that'll be great. So we'll have some time for community participation. And Jeff Taylor is going to facilitate with me. Michael is going to be, and Michael and Jeff are going to be doing the chat. And with us so far, we have Gail Graham, Paul Edmonds, Tom Villa. Nina is going to be showing, Martinez is going to be showing up in a minute. She's having a little bit of computer problems. So why don't we have Gail go first? Tell us a little bit about yourself, Gail. I'm letting you do that because I want you to be able to say what you want other people to know. And then you can tell us about your experiences with the trials.
Hi, everyone. My name is Gail Graham. I am the global representative for Johns Hopkins University AIDS Clinical Trials Network. I'm also the director of Mount Lebanon Baptist Church HIV/AIDS Outreach, and I'm also a patient professor for the University of Maryland Patients Program. I am 25 years plus living with HIV, undetectable, single mom of a 34 and a 36-year-old. So I'm very proud of that. And I also have a 2-year-old Shih Tzu named Ali. So he looks like Baby Yoda, so he goes by Baby Yoda or Ali. So those are my children. My experience with interruptions with the drugs, I did a clinical trial with NIH, and I was very excited because the reason I did the trial was because how can I be on a community advisory board reviewing protocols and not actually, experiencing what it's like being on a clinical trial, especially being in Baltimore and being a part of Johns Hopkins University. Baltimore has a high stigma around research. So I wanted to be able to participate in a research project. So I can go back to my community and say, research has changed from Henrietta Lacks, in this way. So that's why I really joined. I wanted to do a research project. And I'm so happy I did. NIH was excellent. They took very, very good care of me. The doctors were excellent from from when you first got there. Everybody you met, they knew you by name. They sent a cab for me and they treated you so special. But one thing about joining this clinical trials, I didn't really think because I was in a relationship and I made this decision not really thinking about my relationship. And when it came to the part about you had to stop taking your medication, I was fine with it. But my partner, from being comfortable with me being HIV positive, and he was negative, and, being on medication and being non-detectable. And then all of a sudden you got to stop the medication. He was worried. So, we talked about PrEP, and he went to his doctor. When he went to his doctor said, why would you date somebody that was HIV positive? So, that created tension right there. And so I invited him to speak to my doctors. And what ended up happening was we ended up breaking up. He wasn't comfortable with it. And it just didn't work out for the relationship. So I would suggest to anybody who goes into this and they're in a relationship to make sure that they discuss everything with their partner. I would like for the researchers to include mental health for both partners so that as they make this decision and they go through this, they have somebody to talk to when they make this decision. And also to have somebody there to discuss prep with the other partner to make sure that they're okay with that, and that they have some sort of services provided to them too, because it affects them also. But other than that, the trial did not work for me. But the team were very careful, and as soon as my viral load went up, they let me know everything. And so when they weren't comfortable, they said, "Okay, Gail, we're gonna stop the trial and put you back on your meds." But I wanted to really try this a little bit longer. And I said, well, let me talk to my doctors. And, I went to my primary care doctor, and my primary care doctor and the research doctor, talked, and we decided to go for another 2 weeks to see if anything would change. It didn't. My viral load went up again, and I went right back on my medications. But I just love the fact that the team worked together, and they allowed my primary care doctor to, talk to them, and we did it all together. And they followed up with me to make sure I was okay, and my viral load went back to undetectable. The one thing I didn't like, it took maybe 2 years before I got a response back about how that trial ended, the results of that. And it was through a letter. And I thought that the letter was more technical and very impersonal. I mean, I could read it, but if somebody in my community was to get that letter, it would have been trash, because they would not be able to read that. So I think that they need to work on how they communicate the results, disseminate the results after to the community, so that it's they'll understand it. And I think they need to keep in touch with them afterwards a little, to let them know, it's taken a little bit of time, but we haven't forgotten about you. I think that will make a big difference. But all in all, it was a great experience. But I think that for couples, there needs to be therapy, and you need to really think this out and make the decision as a team. Thank you.
Gail, is there anything you think that should have been done differently in the beginning, or is there anything that would have helped you with your relationship, or do you think it was just destined for him to just be too uptight?
I think it was just at that point, because when it's hard to, when you're HIV positive and you're dating somebody that's HIV negative, when it's just hard, period. And then when you involve your own primary care doctor and they say something like that, that really messes with you mentally. And so, whatever he was thinking then, it just, he was just like, I can't do this, because he was going to go back to that doctor. That's his primary care doctor. And he just wasn't comfortable anymore. And so that was just the end of that. And, I was fine. That's his decision. And I was fine with that. I have to respect that. So it is what it is, his loss.
Do we have any questions? Oh, I'm sorry. I'm sorry.
I was looking at what somebody put up there, stigma. I was like, yes, it definitely was the stigma.
Yeah.
And again, I've been living with this for 25 years plus, so it was nothing new. And I developed a shell, so I hate to say that, but.
Any other questions in the chat?
None yet.
Anybody want to pose a live question?
If you do,
you have a live question, please raise your hand, because we've got over 100 people on.
Yeah, that's right. Okay. Well, maybe we should jump to Tom. Tom Villa, tell us something about yourself.
Great, thank you. Good afternoon, everybody. My name is Tom Villa, and I am a community member living with HIV for about the past 25 years. I have been involved with clinical research in various ways over the years. I was involved as a member of clinical trial teams, so the study teams, in previous work in the field of cancer. So I was somewhat familiar with how clinical trials work. And I've been a member of community advisory boards here in the local DC area with our AIDS clinical trials group partners here, as well as with the HIV prevention trials network. I've served on the Ryan White Planning Council in Virginia, and I'm serving on a Ryan White Community Advisory Board here in my local county in Montgomery County, Maryland. And I think most importantly for this discussion, I currently serve on the community team for the HOPE Martin Delaney Collaboratory for HIV Cure Research, as well as on the community advisory board for the RID HIV Martin Delaney Collaboratory. So I mentioned those because those are other ways that we can participate in HIV research or services or HIV cure research, in addition to participating as a volunteer in a clinical trial. In terms of clinical trials themselves, I participated in 2019 and 2020 in an HIV cure-directed clinical trial that involved an analytical treatment interruption. And it's important to know that this was not my first time participating in clinical research. So it's not like I just decided right off the bat, gee, let me do this trial with a treatment interruption. I had participated in maybe 24 different clinical studies or clinical trials over the years that were related to HIV in various ways. And my research team had been asking me for a number of years to participate in a cure-directed clinical trial with analytical treatment interruption. And I had always said no, or at least not yet, because I didn't feel ready. I didn't feel that I knew enough about treatment interruptions and the potential risks to be able to make what, for me, was an informed decision. And a little bit frustratingly, as I considered different clinical trials that they sort of floated to me to see if I might be interested, I would speak with them and read the informed consent forms. And I found that the informed consent forms always talked about potential risks in these theoretical, very vague terms. They would have a category of risk like viremia or viral rebound, but they never included any data, no numbers, no percentages, no findings from clinical research to say, well, gee, we did X number of trials and Y number of people had rebound and it created these problems or it didn't create any problems, no specific data. So for me, that wasn't helpful. And when I would ask my research team for more details and ask specific questions, they would basically just refer back to the informed consent form and basically repeat what was already printed in the informed consent form, which I could read for myself. And so they didn't have any additional information to add to that. And so for me, that just resulted in continuing to say, no, I'm not ready yet because I don't know enough about this. So to summarize or to put a close to this topic, I spent about 2.5 years researching the potential risks of analytical treatment interruption on my own, mainly through working with the various community advisory boards and research projects that I'm involved with. I participated in lots of discussions with various CABs from the Martin Delaney collaboratories like we're doing here, and webinars, some community-led discussions organized by the Treatment Action Group, which I found very helpful to hear from prior participants in trials like that, and a lot of reading and asking questions of community members who had done this before. And honestly, without that information, I'm not sure, I'm doubtful that I would have ever reached the point where I felt comfortable participating in a trial with a treatment interruption, because I just really needed to know more detail than what I was getting from the informed consent forms. So when I eventually did decide to participate, when I felt comfortable enough, I said yes. But the trial I ended up participating in was really my plan B. So my first trial was a gene therapy trial, which I found particularly interesting. And I thought I might be, by participating, might be able to contribute to something really meaningful in terms of new research. Unfortunately, I did not meet the eligibility criteria to participate because at that point in time, I was having some issues with blood pressure and it wasn't sufficiently well controlled for me to participate. So I tried twice. It didn't work out. I eventually needed to take some extra time to get blood pressure worked out. And in the end, my research study team asked me to participate in a trial that they were having a really hard time enrolling participants in. People just weren't interested. And this particular trial was of a combination of two monoclonal antibodies, which would be infused into me to see if they would enhance my immune system's ability to control HIV without taking daily ART. And my overall experience in the trial was good. So I'll give a few details here, and then I'm happy to answer specific questions if that's helpful. So the kinds of things that I thought about when I was deciding to participate were, first of all, what I call the burden of participation. So what's involved? There were frequent clinic visits for about a year and a half. So obviously you need to think about, could I manage that logistically and fit that into my schedule? And I'm single, so for me, I only had to think about myself. But I know from friends and colleagues who have participated in clinical trials that this is also a burden on partners or other family members because it affects them in terms of scheduling as well. My research study team, I felt, did a really good job of sort of mitigating the burden of participation by doing things like mapping out the study visits. They created charts, so like calendar of when I would need to come in, and then a detailed agenda for each visit, what I would do at what time. And they did a really good job of sticking to that schedule in terms of seeing me promptly when I showed up at the clinic. So that really helped to keep things running smoothly, and that was a big help to me, so that things didn't become chaotic. They also offered either transportation to and from the clinic or reimbursement for transportation. So that sort of eliminated that as another thing to think about planning prior to these study visits, which sometimes involve, complicated procedures, et cetera. So that was a big help. And then the final part of this, what I call burden of participation, was thinking about the actual medical procedures, which could be a little complex or for some people uncomfortable. Things like leukapheresis, where they take blood sample, they basically hook you up to a machine and run your blood through a machine, and they select out certain cells they're interested in looking at, and everything else is infused back into your body. Lumbar puncture, spinal tap, if you will, infusions, things like that. So those were the things that I had to consider. Then there was also the potential risks of stopping my antiretroviral therapy. So one of the risks is viremia, and Gail mentioned this already. Virtually everybody is going to have viral rebound in these early trials. The interventions that we're using or testing are pretty early stage, so virtually everybody is going to have viral rebound. The question is, will it be delayed a little bit beyond what would normally be expected? And I think that in terms of managing these risks, current studies, again, are designed really thoughtfully. So the treatment interruptions are no longer than they need to be to answer the question that the researchers are trying to answer. And they have you come in typically every week or sometimes 2 weeks for clinic visits and lab work called safety labs to make sure that everything is being done safely and there's nothing that indicates any unnecessary risk to me as a participant. I wasn't particularly worried about viral rebound because I'm fortunate. Have never developed over the years. I haven't developed drug resistance to any ART. And so I was confident that once I restarted treatment at the end of the ATI, that my HIV medications would enable me to come back down to viral suppression, to be undetectable. And even if they didn't, that there were other options that I could switch to. So for me, that was not like a major risk or concern. But for other people, it might be a greater concern. If somebody's developed drug resistance to certain classes of drugs over the years, that might be something to think about carefully and obviously discuss with your doctor. For me, the greater concern was the potential to further deplete my immune system, my CD4 counts. That for me was the bigger deal because I wanted to be sure that once I restarted ART, that my CD4 counts would either be at or return to quickly my normal levels these days. And I wasn't really willing to take big risks with that. So I was watching very carefully along with my doctors. There was also, as Gail mentioned earlier, the concern about the potential to transmit HIV during the treatment interruption while I was off medications. And again, for me, as I mentioned, I'm single. So I decided that during my trial participation, that abstinence was going to be the way for me to sort of simplify my life and not have to worry about the risk of passing HIV unintentionally to somebody else. That worked for me at the time I participated in this trial. But I think going forward, as these clinical trials involve larger numbers of participants, and the treatment interruptions may in fact need to be longer in some cases, not only months, but perhaps even years, that we need to have better options than abstinence, or just reminding people to use condoms because we know condom use is very low. Because abstinence is not a realistic option for many months or years for many people. So similar to what Gail mentioned, I recommend that there be more supports around not only the participant, but also with their partner available or made available. Things like help in disclosing status and ATR participation to partners. There are recommendations for partner protections, which I won't go into great detail here, but things like making sure that PrEP services are made available to participants and their partners. But if there are questions, I'm happy to answer those, and we hopefully will have time at the end to talk in more detail about partner protections if people are interested. And then finally, the two risks that were not mentioned at all in the informed consent process were the risk of reactivating other latent viruses while I was off my HIV medications. So, for anybody who has other co-infections with hepatitis or herpes simplex virus, which many people have, or human papillomavirus, those can become reactivated if your immune system is stress fighting HIV while you're off meds. So that's a consideration. And then the other thing was potential long-term risks from the treatment interruption. Those were not discussed. And I think as a research community, we can do better there and need to think about that. To end up, my trial involved 2 treatment interruptions. The first one, I had a viral rebound to about 3,600 copies. And as soon as I restarted ART, within a month, I was back down to undetectable. During the second ATI, that was actually interrupted by the start of the COVID-19 pandemic, and all the clinical trials got suspended. So I had to restart ART early, or I was told to restart ART early. And again, went right back to undetectable. And so everything went smoothly there. And then for the end of the study, similar to Gail, I think that there could have been some things done. My study team was great following up with me, but there could have been some things done a little more smoothly in terms of following up with results of the study. They do take time, but I think that it's an opportunity for the study team to keep us participants, us volunteers engaged. And we have a right to know what was learned from our participation. So I think I'll end there. but happy to answer questions. And again, we can talk about partner protections in detail if folks have questions at the end.
That was
Hey, Tom.
great, Tom.
This is Nina. I have two questions. One, what is also for Gail? What is the threshold that they set for viral rebound? And secondly, related, how do they decide, I guess, that your regimen, do they do genotype testing to make sure that your regimen will still work, or how does that go?
So for this particular trial, there were multiple criteria for restarting ART. So the upper limit for the viral load was aimed to be 1,000. So if viral load rebounded to 1,000 or more copies and stayed elevated for more than 4 weeks, then that was one of the triggers, if you will, to restart ART. There was a second trigger if my CD4 count went below 350 or decreased by 30% versus what it had been prior to starting the ATI. That was would be another trigger to restart medications. And then there were others, if there were any kind of like medical complications, or if I decided I wanted to restart, or my doctor decided that I needed to restart. So there were multiple factors. And I'm sorry, there was a second part.
How did they determine that you were okay to go back on the regimen that you were previously taking? Do they do genotype testing in the interim?
They did not. So I think with the current medications, they have a protocol for having you stop your medications. There are certain combinations of medications that you can stop cold, and they don't lead to resistance. And that most of the modern ones are like that. There are some combinations of medications where you have to taper off certain parts of them more carefully. That was not my case. So in my case, we just restarted my normal medication. If I had not achieved viral suppression, then they would have gone into additional testing to see why and to figure that out.
I appreciate the threshold for 1,000 copies is aligned with the WHO standard for viral suppression of 1,000 copies.
This was before all that, and I think
it's been that way for some time for WHO.
So I think that the 1,000 copies was really related to findings from prior research on ATIs and just trying to minimize any potential risks. I was willing, as I mentioned, to tolerate a higher rebound. I was much more interested or concerned in watching the CD4 count. The viral load rebound was less of an issue for me, within reason, so for whatever it's worth.
Bill Freshwater, you have a hand raised, and then Sheck and I see you after Bill.
Hi, thank you. And thanks, Gail and Tom, for sharing. I was on an ATI back in 2018, and as somebody who's been positive for a really long time, back to the days of AZT, I'm just wondering if either of the two of you had difficulties getting back onto your art routine. I mean, I was somebody who was very used to always taking pills, and I was surprised and stay surprised to this, years after my ATI, how difficult it was for me to get back onto taking pills every day. And as these ATIs get longer, how are we going to address that issue? Because it was a big one and remains a big one for me. Thank you.
Yeah, go ahead,
sorry.
Gail. No, go ahead.
When the first trial I was on, I went back to my pills, and it was easy. I didn't have any complications with that whatsoever. I did go on another trial, which was treatment interruption also. But at the end of that trial, it was Cabenuva. I did the Cabenuva, and they thought that I would have a wait to get on that because of some issues that they were having. But thankfully, with the clinic that I was in, within the next month, when I was due for my next shot, I was right on time. So I had no problems. I didn't have to go back to pills, which they thought I would have to do because they were having issues with getting things cleared. But I didn't. I went straight to the 2 shots every other month. So that wasn't a problem whatsoever.
Tekana,
Yep.
Hi, everyone. Shakina here.
Tina.
Thank you, Tom and Gail. I'm curious if during your process of getting informed consent, in the documentation as well as verbal, was there any discussion around while you were on treatment interruption, and just not only just discussing what how you would engage in a sexual practice, but in terms of like HIV criminalization, was there any conversation around, okay, right now in the state that you live in, these are part of the considerations that you need to consider as a part of your decision-making process so that you are fully informed around the possible criminalization of being in the trial itself and, you know, being possibly, this is a conversation I think that we should all be talking about as we progress, because you're right, condoms aren't being used and are ineffective. The FDA has come out and said that. So that's a conversation I would like to be a part of. Thank
Great.
you. And any comments you have?
I would say excellent questions. In my particular clinical trial participation and my cure trial participation, no, those were not part of the process. The partner protections, if you will, that were part of the discussion were really focused on a requirement that if I were going to have sex during the treatment interruption, that I would use condoms and my partner would also use condoms. But as I mentioned earlier, you know, that's not really sufficient these days. There are other considerations that need to be considered, as you mentioned. Gail mentioned also the wisdom, the need to have counseling. So counseling around safer sex, counseling around risk reduction, if you will, which includes not only handing somebody, in my view, and in the recommendations that a group of us, including Gail and Bill and Jeff and others have been involved in, not only handing someone a list of PrEP providers, but also linking their partner to the providers and making that whole process easy and seamless so that it's not just handing someone a piece of paper and expecting them to figure out how to navigate PrEP. And the criminalization issues should also be part of that discussion, and pointing people to credible sources of information for whatever state they happen to live in. I live in the DC area, so people who participate in clinical trials here have to consider not only their home state, like for me is Maryland, but DC and Virginia as well. So because they're all right next to each other. So it can be complex, but there are sources of information. And again, a group of us have worked with NIH to make some recommendations around this, which we are advocating to make a standard part of these kinds of protocols.
Shakila Shaw, do you have a question you wanted to ask?
Hello, how are y'all? I'm gonna piggyback off what Miss Gail said. When I was in the trial for Cabenuva, my body rejected it, and my viral load and T cells went down and through the roof. But they had to go back and do a regenotype. Is that what it's called? To see what else medication I can get back on and get back undetectable. So they did another trial on me, and now I'm on Bictari and I'm back undetectable. And my viral load is undetectable. So, and my CD4 is 864. So it took me like 3 months to get back undetectable.
Are you willing to share a few of those? Are you willing to share a few of those CD4s?
And another thing, I was working where I still work, but I relocated to Deltona, Florida. With the University of Miami, I was a community consultant that we went out in a partner with local businesses and did community engagement, and we did prep and PEP, which was a good turnout because a lot of people were interested in it, and we were able to link some women to care.
So maybe we should proceed now. Let's go to Nina. And thank you. I don't think I said thank you to Gail for telling that really personal and important story. And to you, Tom, for all that information. And so let's move to Nina, who is a lifetime survivor. Nina, you want to tell us what that means and a little bit about your experiences?
Sure. I think the folks who are leading these term redefinitions are still trying to come up with a consensus for lifetime survivor, but what we seem to agree on is that we have the unique experience of having a childhood with HIV. So I am one of those quote unquote rare transfusion acquired cases from San Francisco in 1983. I am now 40 years old. I have the unique distinction of being the first person living with HIV to donate an organ to another person living with HIV, which has been federally allowable under the HIV Organ Policy Equity Act of 2013, which turns 10 next month on November 21st. Lots of people don't know that you can be an organ donor. Please don't remove your registration, keep it. You may be able to help somebody someday with that gift of life. For more information, there is a paper in the Lancet Regional Health Americas, open access, anyone can read it, it's free, the August 2023 issue entitled Living Kidney Donors with HIV, Experience and Outcomes from a Case Series by the Hope in Action Consortium. I am the paper's second author, and I have 2 other successors, so there are 3 living kidney donors with HIV in the United States and 5 more globally. But for purposes of this webinar, we're talking about HIV cure research. I had participated in HIV clinical research for 16 years before I donated that kidney. And late last year, early this year, I decided that before I turned 40, I would like to dip my toe in a little bit of HIV cure research. Full disclaimer, I serve on the Community Advisory Board for RID HIV. And that is the clinical study in which I participated in. I didn't have any analytic treatment interruptions. I don't have a spouse or children. So much like Tom, I just had to concern myself with getting through the process. I did see a lot when I was doing the living kidney donor evaluation. I heard from other living kidney donors whose social support systems had lots of strong opinions about people donating kidneys. And I see a similarity here where other people who are not participating in the research have strong opinions or discomfort. And I will say that for anybody considering participating in the study, get your people in, get your social support into your appointments as early as possible. This is the sort of thing where one-on-one education from you, they may have like a blind spot and may not like hear what you have to say, but if there's a third party, the person who's a part of the research, who's unbiased and can talk to them, the more room that there's available, that can help increase the comfort. And it's also helpful just to have a third person, if you have that type of social support, who can listen to things that you yourself, because as the patient participating in it, you can get a lot of information that can be quite overwhelming. So it's always helpful to bring someone anyway, someone who can listen to things that you may have missed or forgotten and things like that. So for RID HIV, it's really just kind of a sample collection. I felt that with my treatment history with HIV, I've had it for 40 years. I was late diagnosed. I wasn't diagnosed till I was 8.5. I have only had treatment experience with NRTIs and NNRTIs. I spent 10 days on a PI, but that was it. I actually went back to my former triple NRTI regimen for some time, and I actually didn't start and adhere to HAART until 2013. So I've only had 10 years of HAART. And and just not a lot of, never an integrase inhibitor or the rest of the classes. I kind of stopped paying attention after protease inhibitors, but I knew that me clinically was quite a unique case. And even though I was looking to step out of clinical research, I was like, I have not done an HIV cure study. For me, it is kind of hard to find one that I qualify for whatever reason. But I was able to find one here locally in Atlanta, and I said, you want inside my lymph node. I know you do. So for RID HIV, there were like 3 components to the study. One is the leukapheresis part, where they're interested specifically in white blood cells, and leukapheresis allows them to get a large volume without taking a ton of blood from you. I am not a very large person. One of my first clinical studies at NIH was leukapheresis. I did not tolerate it well because I only weighed 98 pounds. I weighed pretty much nothing as a 19-year-old. And so going into this current study, they didn't have pediatric equipment to do the procedure. So they said, you know what, we're just going to do a large volume blood draw. And that's basically, they take a ton of blood so they can separate the white blood cells out, but they have to take a lot in order to get the amount of white blood cells they need. It's an alternative way of getting to the same purpose. So they drew a cup of blood. I went home, took a nice nap. I earned it. And then the 2 other parts of the study were fecal collection, which wasn't a big deal. They make cute little kits. It's pretty nondescript. And I've done 2 of those for 2 different studies. And then the last part is invasive. It's the lymph node biopsies that you've probably heard from other studies. And that's where I sort of ran into, I guess, some expectations over, did I sign up for this? I knew the lymph node resection is itself straightforward, but I would say that if you're considering doing something like that, make sure that you do have social support at home in case complications do come up. I did run into the complication of being allergic to the skin glue that they used. So if you're consenting to a lymph node biopsy, there is a chance, and they say it in the informed consent, that you may be allergic to the skin glue. They could have done a little bit more education about what that might look like, since the incision is covered, you can't see it. So I wasn't really aware that I was having an allergic reaction. I just knew that I was in a lot of pain, and it took the surgeon 9 days to say, send me a picture. And then he says, oh, you might be allergic to the glue. That will explain some of your pain. And then I think by day 10, I just couldn't take it anymore. So I removed the glue myself. And I made sure, I said, look, having a post-operative day 2 call is not sufficient follow-up. These people are volunteering to let you take, they're technically organs as well, organs from their body. The least you can do is to make sure that they're healed up by day 14. So I know that the protocol has been amended to include additional follow-up for all participants. And then to have a more robust surgical conversation towards the beginning of the study, because I think same-day surgical consents for research can be a bit coercive. I think if we're acknowledging that people living with HIV have cognitive issues, that, you know, having a surgical consent the same day that the OR is booked, and you're saying, do you have any questions? Or you can say no, it's not quite the right environment for objections. And I know that some people are like, well, Nina, if you share all this, you might dissuade people from research. No, I think it's educational to disclose what happens. I think that people who want to give, nothing worth doing is easy. And it certainly doesn't dissuade me from future research. I'm just kind of at the end of my 20 years. I've certainly given enough. And moving forward, I think if I'm participating in clinical research, it'll be when I turn 50 in 10 years, or if there's a potential clinical benefit. But I think it's just fair and honest to be transparent. Like, we can't always give shiny, glossy examples, and not everything can be covered in the informed consent, but you can always ask questions about products that they'll be using, types of anesthesia, and what are your preferences for anesthesia? I did try to say, you know, I have no social support at home. I would like to forego to midazolam because that lasts all day. I'm okay with propofol. It's short acting, and my wishes weren't honored. I'll say that. But there are things you can do to advocate for yourself. And I raised all these issues with the research team. There were copious emails. and that they have honestly like definitely changed it so that more patients moving forward don't have to, if something happens to them, they'll be better supported.
Well, Nina, that was really helpful and interesting. Thank you so much. Any questions? Anybody from the chat or live questions? Got a few minutes. See anything, Michael?
No questions.
So, Nina, let me ask you, hopefully you're so I so understand that you need a bit of a break, but you're still willing to participate in community forums like this, right?
Well, yeah, people don't get funded to do community dissemination of research, and I do think that that's very important.
Yeah, because I mean it's so important to hear your voice, because your community is one that so many of us don't know enough about, and it's been quite an eye opener and an earful and a brain full of information that we've all learned from your participation. So we love you and hope you will hang out with us.
You can pick my brain, just don't biopsy it.
There you go. Fair enough. Fair enough. Funny. So any other questions for Nina before we go on to Paul?
No, there's just been a lot of support and thanks in the chat.
Oh, that's great. That's wonderful.
I do see a question for Shakina Rose.
Okay. All right. Go ahead.
Go ahead.
Nina, I don't know how to phrase this. It comes from a good intention. So please, if I offend, know that it's not meant for that in that way. So, recently there's been a big discussion around HIV reservoirs. And I'm just curious if you have any kind of thoughts around how you may experience that, because you experience living with HIV uniquely, right? We all identify that, right? So I'm just curious if you have any thoughts about this emerging field, and how can we support you?
I mean, I don't know that that's what participating in the RID HIV study will illuminate is what they find in my lymph node in that reservoir any different than someone who may have acquired HIV later in life. I certainly think that having it from such an early age and since before treatment was available is kind of a benefit. I feel supported by the HIV community. I think lots of discussions that folks have in the community, I don't necessarily find are applicable to me. Like the idea that, for example, I am actually asexual and aromantic, and that has nothing to do, I don't experience romantic or sexual attraction, has nothing to do with HIV. I know I'm fine as beep, and I could have a relationship. It's just, for me, sex is great, but have you ever beeped the system? I love that. I've lived for that. There's just a way that I do navigate the world that is pretty unique. And I like to say that my heart is actually too big for one person. So a lot of the social hierarchies and constructs that a lot of people living with HIV ascribe to, I actually don't. There's a lot of community support for campaigns that prioritize the importance of treatment, that actually focus on somebody who's not living with HIV. And I have always centered people living with HIV. I love to be on treatment because it's good for my health. And I'll say that living in Georgia in 2023, my friends who die of Kaposi sarcoma, gay Black men in the South, were having sex just fine. But, being on treatment didn't fix what broke inside them. So, in those ways, I think when we're looking at popular messaging, we need to consider the source. We need to consider who's being left out. And I think this year's U.S. Conference on HIV/AIDS, the folks who started the Dandelions movement tried to remind people that, yes, people who acquired HIV also in the 1980s are also aging with HIV, but people forgot about us because about 90% of us died. So just remember like who we are leaving behind and those who we aren't connecting with, because if we connect to them and we lift them up, it lifts everybody up.
There was an interesting question here in the chat by Julie Patterson, who was asking if the panelists that have shared so far have noticed a difference between the trials that were sponsored by Big Pharma versus trials that were sponsored by a network, an HIV network. Have you noticed any differences between the types of studies that you've been involved with? Go ahead, Tom.
Yeah, thank you, Michael and Julie. I'm a slow typer. I was trying to respond to you in the chat. So I've participated in just one industry-sponsored trial, and that was of a candidate or an experimental vaccine against the Ebola virus, which I decided to participate in because the Ebola virus is endemic to West Africa, where we all know that HIV is also present. And so I knew that any vaccine would have to be shown both safe and effective in people living with HIV for it to be useful in the real world. So that was the reason why I volunteered for the trial. I wanted to put my virus to work. I found the trial was extremely well run. And I think that the industry trial actually did a better job than any of the other trials that I've been in terms of the informed consent process. They not only had the printed informed consent form and the one-on-one review of that and discussion with a member of the study team. Actually, it was the PI, which was better than most of the other, more direct than most of the other trials I've been in. It was the actual PI who met with me, the principal investigator. But they also had a video-based educational program that we watched, that participants or prospective participants watched in small groups. There were maybe 10 or 12 of us together who watched this video as a group. And then we had facilitated time for discussion and questions and answers. And that was prior to going through the informed consent process on an individual basis. So I thought that they did a really good job, a very professional, well-done job of presenting complex information in different ways, different media, different styles that could appeal to people who may learn differently, not just from reading a really complicated informed consent form. So I really like that, and I would love to see the networks, the clinical trials networks, replicate that kind of an approach. I understand that it takes some money to create those kinds of things, but there's money in the network. We could do multimedia material like that on general things that are common across all of these trials, like what is an ATI. We could create that at the network level, and we don't have to create it individually for each trial. So there's a way to do it, and I would love to see that. It was very well done.
Okay, so let's jump to Paul. So Paul Edmonds is the City of Hope patient and has a very unique experience, not only coming out to the community, but to the whole world to let us pick his brain and body about being cured from HIV. So Paul, you want to tell us about your experiences?
Sure. Thanks for having me here. Good morning, good afternoon. Anyways, I'm 68 years old. I have a husband. We're almost our next anniversary will be number 32.
Wow.
So I tested positive for HIV in 1988, and also received an AIDS diagnosis because of my low T cell count being under 200 at the same time. So my first drugs were AZT, although they had cut the dosage in half when I started. So I'm really glad that happened. But I tried everything as it came along. I was in quite a few trials. Everything was going great. Then in 2018, I had been going to see my HIV doctor for years, every 3 months, and they always did labs. And suddenly my labs just all went haywire. So I went to a hematologist, and they discovered that I had MDS, or myelodysplastic syndrome. Uh, and they wanted to start chemo right away and told me about a bone marrow transplant. Uh, I decided I was planning to move to Palm Springs anyways in the near future. So I decided to go to City of Hope, which specializes in bone marrow transplants. And I spoke with them and decided to hold off on starting the chemo while they looked for a donor. And they told me they would look for a donor, that had this rare mutation that prevents HIV, the CCR5 delta 32 mutation. And it was about a month after seeing my doctor there, they called me up and they had found a donor. So I came on down to Palm Springs and went into the hospital at City of Hope in October of 2018. And they started me on chemos to get the leukemia in remission. And that took 3 different kinds of different chemos and some pills and different things to get it in remission. Took about 3 months. Then they did the bone marrow transplant February 6th of 2019.
This is really interesting.
Yeah, I can tell.
No, yes,
This is actually interesting.
this is the guy that was cured of HIV.
Okay. Oops.
If you're not speaking, could you mute, please? Thank you. Well, at least you know you're interesting, Paul. We
Now,
knew
I kinda
it.
kinda lost
We
my
knew
place.
it. Go on, sweetheart. Sorry.
So I'm up to the bone marrow transplant was February 6, 2019. And the plan was to stop my HIV meds a year later. And then the pandemic hit during that, right at almost at the end of that first year. And I was really nervous about that. I was more nervous about that than stopping my HIV meds. So I waited until there was a vaccine available. So I ended up starting the stopping the HIV meds about 2 years later, and that was on February or March the 2nd in 2021. So I'm approaching 3 years without the HIV meds. Next March will make 3 years and 5 years for the transplant. So I was ready for my ATI when it happened. City of Hope were just wonderful. They sent someone to my house once a week to do labs. So they kept a very close watch on me. The first 12 weeks was once a week. Then they went to every 2 weeks for the rest of the year. And they still are following me, and they'll follow me the rest of my life. So, I was not nervous with them at all. I trusted them. And their informed consent was very, very easy to understand. And they really went over it well with me. I can't say enough good things about them. They did a fantastic job and continue to. So I don't know what else can I. That's it in a nutshell.
Well, that's a mouthful. Does anybody have any questions? Anything in the chat? Anything you'd like to ask Paul?
Yeah, again, please use the raise hand function, as we still have over 100 people on the call, and that way we can recognize you and have you ask your questions. In the meantime, I'd like to, and I'm scrolling back to it, Ron Ellis had an excellent point in the chat that, let's see if I can find this. It's earlier on.
So, Paul, while he's looking for that, how often do you have to go to doctor appointments now? Are they as frequent, less frequent, kind of in between?
Yeah, they're less frequent. I went 6 months with seeing my hematologist, but then some things, little things pop up. So now it's back to 3 months. It started out I was going in once every 2 weeks. Then it eventually went to once a month, then 3 months. 6 months is the longest I've been, and that's only happened once. But I'm back to 3 months right now. So. it's an hour and a half away on a good traffic day to go to City of Hope.
Ron's on the line here. You have your hand raised, Ron. Do you want to share?
Yeah. Jeff mentioned my item in the chat, and it was just that, Tom has an incredibly good point about providing more detail about the extent of the risks of whatever is being done in a clinical trial or even in observational research with things like lumbar punctures and lymph node biopsies. And so that information is available, and study leads should be willing to share the study protocol. It could be either the IRB application or the improved entire study protocol, so that people can have access to that more detailed information.
Nina, you had your hand up, and then I see Tom, you have your hand up.
Yes, I'll just piggyback off that comment. I think to some extent you can standardize the kinds of knowledge that people need to know, but at some point it really does become personal and individualized. And for example, yes, kidney donation, there's lots of information about it and what's expected and things like that. But a lot of people's journeys in doing something like that is very individualized to medical history and then also at the transplant center. So I think there's a similar case here. It's just, do you provide as a research site room for people to articulate where things might be heterogeneous or things that need to be of concern? Because I think for a lot of research sites, they think that IRB approval means that there's nothing wrong with the study and it's good and there shouldn't be any more pragmatic considerations. And we all know that that's false. But my actual question to Paul was, did you do anything celebratory for both the cancer remission and the cure from HIV? I personally went on a kidney moon when I punted the kidney, since I'm not married or have children, but I did a little something for myself. So I was curious if you commemorate in any special way.
Well, I certainly do. February 6th, the day of the transplant, is like my second birthday. So it's kind of my first one now, but I've still been planning to have a party to celebrate everything. I haven't gotten it together yet, but soon I hope to do that.
And Tom, did you want to ask?
Yes, thank you. No, I wanted to respond to Ron. Thank you for your response. I guess my view was in part a reaction to the informed consent form and the fact that the form went into what I felt was significant detail on the risks of things like venipuncture, blood draw, and other sort of routine procedures that, there'd be a whole paragraph about that. And there was no detail at all regarding potential risks of ATI. A sentence or two sentences, but there was a paragraph about venipuncture. So I thought that was sort of not proportional. And I don't think that we need to go into, all of the science and research, but I think that if we want people to feel comfortable to participate, we can help them to feel comfortable by perhaps summarizing the risks with a little bit of data to provide some empirical perspective. What have we found? The risk, is it low, medium, or high for this particular risk, and what does that mean? I think there are ways to do it. It's complex, but I'm not sure that we can expect other potential community participants or volunteers to spend a lot of time doing their own research to figure it out. It's much easier just to say no. So it's food for thought. But I do think there are ways to do it. So thank you.
That's very interesting, Tom. The thing of it is that the docs are getting to a point where, oh, well, anything over 2 pages is too much because people stop reading and it's too much information for them. So, and I remember being on hepatitis trial and thinking, okay, I'm going to get all this information. And it was like 2 pages of nothing, So somewhere there's got to be a happy medium, or maybe two ways of doing that. A 2 or 3 page thing and an additional information sort of thing for people that are interested in the kind of facts that you needed to have before you felt comfortable proceeding. I think if anything, we can take that message home with us after this call. The other thing too is that maybe for Paul, we used to have anniversary parties for Tim at different meetings. So maybe we can continue that. I guess I should just have shut up and done it as a surprise, but I'm not gonna do that. I'm a big mouth. So, I mean, if you feel comfortable with that, maybe we
Oh, I
can
do.
that tradition.
Absolutely. I'd love it.
And that's great.
Right.
That would be great.
This is
So-
Jeff. I'd like to chime in if I could on the
Sure.
discussion about the informed consents. This really speaks to the reason why we need community embedded in the process from the beginning. We need to be on the advisory boards, reviewing these studies as they're being conceived, how the researchers come to us as they're thinking of this and saying, A, is this something people want? Is this something people sign up for? How can we make it appealing to them? And then once they've drafted something, come back and have people review it, so that all too often, the first time people see it is once it's already kind of written in stone. They may have already gone to the IRB and it's too late. So it's really important to have us involved, at the beginning and really looking at these things. I mean, all the stuff Tom was talking about, they have a whole paragraph on the dangers of blood draws, but hardly anything about the ATI, which is the most important part of the study and a lot more risky. That's just copy and paste. That's just laziness. You've got somebody putting this together and they just put in the boilerplate language that's been used in every other study, often including the inclusion-exclusion criteria. So they won't allow people over 50, all this stuff that's just copied from somewhere else. Nobody's thought about it. There's no scientific rationale. So I just, a real call for having us there at the table. All these ideas that we're talking about here could be easily incorporated, but they need to ask us up front, because if they wait until they're ready to start the study, they don't have the money there, nobody's thought about it, they don't have time, etc. It's too late. Thanks. Nina, I see you got your hand up.
Yeah, I was just like, we talk about what it is that people need to have like a better informed experience. I wondered if anyone's done any work on predictors of willingness to participate in HIV cure research. I know that it's been done for HIV vaccine research, and you'll find that, a number of social behavioral theories will predict willingness, one of them being like the theory of reasoned action. And that theory talks about how for some people, personal attitudes and individual beliefs are more drivers of behavioral intent and as a proxy to behavior. But for other people, it would be social norms, and it would be a motivation to comply with those social norms, and to just be mindful that, like, I don't know, we talk about a lot about what research says, but research talks about a group of people. So if 80% of people experience X, that does not necessarily mean that you as a person have an 80% of experiencing X. That's an ecologic fallacy. And so I just want to point out that nuance of either something will happen to you or it doesn't, and you as an individual contribute to those population statistics. So it's just everybody has a different motivator to what prompts them to want to seek more information. And I'm just curious if that's been done for HIV cure.
Well, when we first started this, and there was a lot of work done in this. It's very interesting to compare this with treatment trials for which there's none of this. And there's been social science done around this pretty much from the beginning, because, unlike the treatment trials where people really need, especially in the beginning, needed treatment and care, they kind of need cure participants more than they ever did treatment participants. So we've got Corinne Dubé. I'm not sure she's on the call with us today, but she's been a real hero in this arena and trying to get this done. And actually, I think the first surveys that I remember were done in Europe, and there was a Dutch thing that happened first, and we were like, wow, we should do that here. And actually, we decided when we started this long-term survivor, lifetime survivor webinar series that we were going to do a study of behavioral study in conjunction with this and push one of our agenda items, which is allowing people over 65 to participate in cure trials if they were able to meet the inclusion exclusion criteria and were otherwise healthy, because we don't want any really hideous risks to people that are over 65. And so in conjunction with that advocacy, we're going to be starting a little behavioral study soon that is for people that are over 65. So we can find out their willingness to participate, things that would perhaps prevent them from participating, their reaction to the ATI, the different procedures, all the kinds of things that we've been talking about here that have never been asked of that particular community. So we're really behind this, and social science has been a big part of our advocacy from Jump Street, I think. Karina, are you on the call?
No, she's not.
Oh,
That's
sorry.
after other folks besides younger than 65?
Well, that was because it was a long time, whatever. We wanted to be sure to get we had heard at a lot of different meetings, it's kind of what you're saying, Nina, from your community. What about me? I'm here. Why can't we get involved in this too, or be a part of this as well? So we heard that, and we moved on that for this particular instance.
Right.
But we don't want to tell you this, but we will, I think.
Find in that group, it's not going to be like, it's going to be different across all groups. And I'm just trying to get us in general, if these motivators and predictors of willingness to participate have been ascertained for HIV cure research globally, with the full knowledge that yes, in people over 65, they may differentiate in some appreciable way.
Well, this was a surprise, but I am going to tell you this anyway, that we have been talking very seriously about doing the same sort of work for lifetime survivors when that's finished. So, and I think that you can be very proud that you and your group have really have motivated us to look into that as well. So we'll be in touch. Was one of the reasons that I asked you that hopefully you're not going to disappear, right? So anyway, but we're trying to include everybody. And I think that's something that from the beginning, we wanted to start this cure research differently, to include all communities and to educate all communities about what was happening in this regard, unlike what happened the first time around with treatment.
I see Thomas had his hand up, and then Hulda is as well.
Yep.
Great. I wanted to piggyback on a point that Linda made, which I think is very important for us to keep in mind as we talk about these trials with treatment interruption, and that is that these HIV cure-related trials are unique. So as Linda mentioned, people who participate in trials, let's say cancer patients who participate in clinical trials, are generally participating in trials of new potential therapies, treatments, after they have failed the standard treatments. So for them, this is like a last hope, if you will, to find an effective treatment for their cancer. In the earlier days of HIV, a lot of the treatment trials were of new therapies that were not yet proven. And so again, people were participating with the hope that some of these new therapies would actually work for them, which was great. With HIV cure-related trials, we have the opposite situation. Many people today, particularly here in this country, have access to effective HIV medications, and we can live long and healthy lives if we take our medications and are adherent to them, et cetera. But to participate in these trials, we're being asked to stop our medications in order to help test or evaluate whatever it is that the study is looking at. So we're asking people to stop doing something that protects their health in order to assume some risks to participate, to evaluate a treatment or other thing that will not benefit them at all personally. And right now, these trials are still early stage enough that we should not expect personal benefit from participation. Someday that may be the case, but it's still a little bit early to expect that. So it is a different situation, and I think we just need to have that in mind as we're thinking about participation and what kinds of supports, including the mental health assessments and supports that Gail mentioned for both participants and their partners as they go through this difficult decision, and through the complexities of navigating participation without treatment. It's a lot to think about. So thank you for bringing that up.
Hulda?
Yes, I hear you saying the trials, you have extended the trials to people over 65, but how far over 65? For instance, I'm 79 years old.
So we haven't extended them. We are starting a movement in hopes of having the inclusion criteria changed to include over people 65. You know, a lot of this stuff, as Jeff mentioned before, is just cut and paste, okay? And actually comfort by a lot of times the pharmaceutical sponsors that their drugs, you know, look better in people that are younger, especially in HIV where there's immune issues. So we're starting a movement to try to include people over 65. Might take us a while to get to 79, but we got to start somewhere, right?
That's right. Thank you.
Yeah, to that point, we've been really championing this at the ACTG, the AIDS Clinical Trials Group, where a lot of these trials end up when they're ready to go into people, especially the larger trials, if a pilot proves promising. So every single time we see an age limitation, we ask why. You know, sometimes it's just an arbitrary cutoff. Sometimes it was cut and paste. Maybe there's an actual reason. But what we're starting to demand, as Linda said, is there needs to be a scientific rationale for every exclusion criteria that's going to kick somebody out who could benefit from this. So older people, lifetime survivors who have kind of a different treatment course, you know, women, you know, all too often get left out. So yeah, we're always advocating, of course, people of color, because that's where the epidemic is the greatest. So yeah, we need to remind people to be mindful of why they're doing things, and it's not just kind of this rote, it's what we did before.
Okay.
There was a question to Paul earlier, which was about how he feels like he fits in with the HIV community ever since the cure that he's experienced. If he feels like there's been any changes and any psychosocial needs that are being not addressed for someone who's post HIV or who's been cured.
Right. I mean, I very much still feel a part of the HIV community. I kind of don't know how not to. You know, it's more than half my life that's been my main community. So, you know, I don't feel any changes there. I mean, I just haven't run into a lot of people who, you know, are negative about what's happened to me. Perhaps that may happen in the future, but I haven't had that experience as of now. So I feel very connected to the HIV community. It's my main community.
Great.
So maybe, Michael, it's a good time to put up the evaluation so we can get a gauge of how people like this session, and so we'll know in the future if we're doing what we're doing right and not. So that, and maybe, unless there are any other questions in the chat or any other live questions, maybe we can have Tom take the last 10 minutes and talk to us about the partner protection issues that he had raised earlier. Does that sound like a plan?
This is Jeff. I did pose one question in the chat to Dr. Ellis, who does a lot of neurology research, to get his take on, you know, if he thinks there are any dangers in doing treatment interruptions of making those issues worse or making them come up, and if he has any recommendations on how we should screen for that either before or during a trial.
Yeah, great point, Jeff.
Yeah, I'll be very brief in the interest of time, but I do have thoughts. One is that we just don't really have any idea how the cure interventions are going to affect the brain. And it's fairly routine for the people writing the protocols to not include any measures of neurological function or brain structure or anything other than just a simple questionnaire about the patient. You know, did you experience hallucinations, depression, or something? We know that the cells that harbor HIV in the brain are different from those in other parts of the body, and that the brain has its own special immune system. And so it's entirely possible that the effects on the brain will be very different from the effects on the rest of the body, and it might cause havoc. Now, fortunately, that hasn't happened so far that I'm aware of. But I personally think it would be better to have more, even simple measures of neurological function as part of the studies, part of the main protocol, not just some study, sub-study. Yeah.
Yeah, thank you, Dr. Ellis. It's great points. And as I was
Yeah.
hearing you talk, I'm thinking maybe we need to do a webinar just on that topic.
Yeah, and
Just
maybe
call me Ron for God's gosh
okay,
sake.
Ron. Well, now, Ron, since we're being so friendly here, I wonder if you might give us a few lines or bullets or paragraphs on something on advocacy items that we should ask to be included in these trials.
Yeah,
Would you mind doing that?
sure.
That would be
I can
fabulous.
summarize what I would say, which is brief neuropsychological testing performed before and during the treatment, and then during the phase when virus rebounds or afterwards, so that we can know what these interventions are doing. And there are now some things that you can measure in blood so that you don't have to do a spinal tap that reflect what's going on in the brain. So those things could be added as well. You know,
Wow.
Jeff is familiar probably with NFL, neurofilament light. You can also measure some Alzheimer biomarkers in blood. So lots that could be done that's not being done.
I hope we have the tape. I hope we've taped this, right?
Yeah, of course.
No, great. Great. Great, Ron. We love you. Thanks for coming. And
Nina
thanks
has
for
her hand up.
your tips.
Is that
Nina has her
Okay.
hand up.
Oh, Nina?
Yeah, I just wanted to mention, thank you for putting a little focus on neuro HIV. I did see also Tom in the chat. We've participated in the same NIH study looking at thinking and memory problems in people living with HIV that's run by Dr. Avindra Nath, the clinical director of the National Institute of Neurologic Diseases and Stroke. That study very much focuses on doing neuropsychological assessments, MRIs, PET scans, and my brain is having a perimental pause, but folks like him are very big proponents of focusing on neuro HIV. I know Tom can probably speak to more of completing the study because I was let go halfway through a brain hemorrhage I had as a newborn, for which I received 13 lumbar punctures in the first 2 weeks of life. And on the last day of getting a lumbar puncture, I got a blood transfusion, which gave me HIV. So that's a good time. And but I just also want to mention that Emory is doing several studies with the drug baricitinib, focusing. They've used it in long COVID patients to see if it will help some of their neuropsychological complications of long COVID. But they're planning to launch a study very soon. They just got FDA approval to study this drug in HIV patients, and I'm currently planning to clinically screen for this study because, again, it might confer a benefit for me. The study I am planning to enroll in focuses on inflammation and depression in people living with HIV, and more to come on that front. So yes, I retired from clinical research, but not really.
You might be interested to know that there is a trial of that same drug for cognitive problems that is under development. I think it's getting pretty close. So.
Yeah, I specifically chose the inflammation and depression study because a lot of people don't really know that HIV itself has an effect on the dopaminergic system, and a lot of people out there living with HIV are struggling with dopamine-related depression. They think it's all just related to stigma. It's not. There's a biologic basis, and as somebody who donated a kidney, one of the drugs that's available, it's the only drug available for dopamine-related depression. I can't really take anymore. That's kind of the price I paid a little bit for science. And so I'm looking to this study as a hopeful alternative.
Well, hopefully you'll get some benefit. Wow, Nina. Ugh, every time I learn more, it just is head spinning. Okay, well, Tom, we only have 3 minutes. maybe, maybe the best thing to do is, do you have a link to all of the stuff that you've written about this? That might be the best way. I'm looking at the survey now. Let me close this. Way great. 80% very satisfied. We love that. Thank you for voting.
So Linda, I put in the chat this paper by Corinne Dubé and a team of co-authors. It was published in 2023, so earlier this year, in Lancet Infectious Disease. And it talks about a framework for partner protections during HIV-related clinical trials with analytical treatment interruption. And it includes detailed discussion and recommendations from this group for partner protections, as well as for the psychosocial readiness assessments and supports for ATI participants and their partners before, during, and after participation. So for anybody interested, that would be the source, if you will, of this information. But we could also schedule another webinar in the future if this is something that people would like to discuss in more detail, if they find it helpful.
Thank you, Tom. And thank you, Gail, for your personal experiences. Tom, for all that you've been through and all your advocacy. Same with Nina, who continues to be one of my heroes. And Paul, thank you. We're all so happy and hopeful to hear you, see you, think about you, and now to celebrate with you, we hope. So thanks, Michael and Jeff, and all the people that have made these sessions just so wonderful and rewarding for us that I've tried to put them together, because it's very rewarding to know that you care about this and are interested in this. So thank you for participating. Our next scheduled session is at the same time on November the 21st, and that will be Where Do We Go From Here? It'll be very similar to our panel discussion this evening, wherever you are. And morning, I guess, is where most people are besides the afternoon. But anyway, to see what we've learned here and what we can carry forward in our everyday advocacy, what we should ask for, what we should try and demand. So thank you so much again for participating, to our panelists and to all our attendees. So hopefully we'll see you next month. Jeff, do you want to say something before we go about the last gift webinar that we're going to have? It's going to be it's going to be next year, but next year is
January.
really
I don't have the date in front of me, but we will send it out to everybody on this list. Last gift is a study that many of us have worked on, an autopsy study, actually, so we can get those brain biopsies that Nina was talking about. So it's a really fascinating project. So we look forward to sharing that with you in January. So check your inboxes.
Yeah. Just wanted to give it a little preview. So there's more to come. So stay with us. Stay tuned, as they say. And thank you again to everybody. Thanks to you, Michael, as well.
Here, everyone.
Thanks to you, Linda, for being our hostess with the mostest.
Thank you. Sorry for the earlier noise.
Thanks, everyone.
All right.
Thanks, everyone, for putting this together. Bye for now.
You're welcome. Thank you. Thanks
Great
again
to
for
see you,
coming.
everyone.
Bye-bye.
Thanks all.